The title of the paper is Simulating 500 Million Years of Evolution with a Language Model. This research presents groundbreaking work in the field of protein modeling and evolutionary biology. The authors of the paper are as follows: Thomas Hayes, Roshan Rao, Halil Akin, Nicholas James Safroniev, Denise Oktay, Ziming Lin, Robert Verkel, Vincent Kwai Tran, Jonathan Deaton, Marius Wigert, Rohil Badkundri, Irham Shafkat, Jun Gong, Alexander Derry, Raul Santiago Molina, Neil Thomas, Yusuf Khan, Cheta Mishra, Carolyn Kim, Liam J. Barty, Patrick D. Shu, Tom Surku, Salvatore Candido, and Alexander Rives. More than 3 billion years of evolution have produced an image of biology encoded into the space of natural proteins. Here we show that language models trained on tokens generated by evolution can act as evolutionary simulators to generate functional proteins that are far away from known proteins. We present ESM3, a frontier multimodal generative language model that reasons over the sequence, structure, and function of proteins. ESM3 can follow complex prompts combining its modalities and is highly responsive to biological alignment. We have prompted ESM3 to generate fluorescent proteins with a chain of thought. Among the generations that we synthesized, we found a bright fluorescent protein at far distance with 58% identity from known fluorescent proteins. Similarly, distant natural fluorescent proteins are separated by over 500 million years of evolution. The proteins that exist today have developed into their present forms over the course of billions of years of natural evolution, passing through a vast evolutionary sieve. In parallel experiments conducted over geological time, nature creates random mutations and applies selection, filtering proteins by their myriad sequences, structures, and functions. As a result, the patterns in the proteins we observe reflect the action of the deep hidden variables of the biology that have shaped their evolution across time. Gene sequencing surveys of Earth's natural diversity are cataloging the sequences and structures of proteins, containing billions of sequences and hundreds of millions of structures that illuminate patterns of variation across life. A consensus is building that underlying these sequences is a fundamental language of protein biology that can be understood using large language models. ESM3 stands out as a multimodal generative language model that simultaneously processes and generates data across 3 distinct modalities: sequence, structure, and function. The architecture operates primarily as a bidirectional transformer, which allows it to analyze context from all sides, both previous and subsequent tokens, when making predictions. ESM3 is trained using a generative masked language modeling objective, masking portions of input data and challenging the model to predict them based on other tokens. The largest variant, trained on a dataset of 2.78 billion proteins with 98 billion parameters, achieves remarkable representational fidelity for sequence, structure, and function. The ability to issue complex prompts to ESM3 opens up an entirely new dimension in protein engineering. By specifying a combination of structural and functional requirements, researchers can guide the model to produce innovative protein designs tailored for specific applications. The authors emphasize that ESM3 operates as a sort of evolutionary simulator. While traditional evolutionary processes are painstakingly slow, requiring millions of years for beneficial mutations to arise, ESM3 can generate functional proteins quickly, allowing exploration of distant expanses of protein sequence space. Today, we embark on a fascinating journey through the world of evolutionary biology and artificial intelligence. We're delving into groundbreaking research that showcases how advanced language models can simulate hundreds of millions of years of protein evolution, all while generating functional proteins that are remarkably distinct from known sequences. Protein language models, like ESM3, harness the power of natural language processing techniques and apply them to biological sequences. These models are trained on vast datasets comprised of protein sequences, allowing them to learn the complex relationships between amino acids, the building blocks of proteins. A key innovation in ESM3 is its multimodal capabilities. Unlike traditional models that focus solely on sequence data, ESM3 integrates not only the amino acid sequence, but also the 3-dimensional structure and predicted functions of proteins. This holistic approach allows it to reason across multiple dimensions of protein biology. The training process for ESM3 is incredibly complex and resource-intensive. The model was trained on a dataset containing 2.78 billion protein sequences, leading to a staggering 771 billion unique tokens. The training process involved data collection, masking strategy, and careful use of various techniques to enhance model performance. ESM3 stands out as a multimodal generative language model that simultaneously the architecture prediction. ESM3 is to now let's explore a critical among numerous indicates it is crucial looking ahead. The authors suggest fine-tune. In summary, Figure 1a illustrates the iterative sampling process of ESM3. ESM3 begins with a sequence of masked tokens, representing an initial state for protein generation. At each time step, the model samples masked positions iteratively while considering context from previously generated tokens. This process maintains coherence in the generated sequences and facilitates realistic protein folding dynamics. Figure 1b presents an overview of the ESM3 architecture, highlighting the flow of information through input tracks and the processing pipeline. It emphasizes the integration of multiple modalities, sequence tokens, structural tokens, and functional keywords. Input tracks are converted into embeddings and fused into a single latent space for processing. The process of residue, encoding both orientation and position in three-dimensional space. The output is a sequence of structure tokens that encapsulates essential geometric relationships,